Hello, I'm so honored to be here to present one of my previous cancer studies. I'm Associate Professor in the UCLA School of Nursing. I'm Adjunct Associate Professor in the Department of Epidemiology at the UCLA Fielding School of Public Health. I'm a faculty member of the UCLA Johnson Comprehensive Cancer Center and the Human Genetics Department of School of Medicine and the Genetics Genomics Graduate Program in Bioscience. I'm a molecular genomic cancer epidemiologist. I got a PhD degree in the epidemiology at the University of Pittsburgh. During my graduate studies in Pittsburgh and later, I accumulated an array of research experience in the hospital-based multiple cancer cohorts for cancer treatment and prognosis. I completed my postdoctoral fellowship in the Department of Epidemiology at the University of Texas MD Anderson Cancer Center. My research here at UCLA has focused on examining cancer-related metabolic and inflammatory biomarkers associated with lifestyle factors as cancer prevention and strategies. My current genomic and epigenomic studies evaluate interplay of environmental behavioral factors and genomic epigenomic markers on molecular pathways leading to cancer carcinogenesis and progression, which will contribute to the development of genetically and epigenetically targeted preventive intervention. A brief background. Several DNA methylation studies for breast cancer initiation and progression supports global hypomethylation and focal hypermethylation, such that some tumor suppressor genes are frequently hypermethylated as CpG islands and promoters, so they are inactivated. However, the role of epigenetic mechanisms in the breast cancer tumorigenesis has not been conclusive. For example, there is no consistent trend towards an association between identified CpGs and the risk of breast cancer development across different studies. These suggest the need for larger population-level epigenetic studies which prospectively evaluate breast cancer development, specifically in breast cancer molecular subtype stratifications. In particular, among postmenopausal women, the obesity-insulin resistance connection is well-established factors for breast cancer risk and progression. However, their interconnected molecular pathways on the DNA methylome have not been well established. Aberrant DNA methylation may directly or indirectly influence the function of pancreatic beta cells as well as other organs involved in the glucose homeostasis. Also, even a modest change in DNA methylation causes a substantial effect on gene expression, and in the late onset disease, DNA methylation has a large effect on disease development over a long time period. So DNA methylation can serve as a biomarker to detect an early at-risk group for morbid conditions such as insulin resistance and breast cancer, even several years before the clinical diagnosis is made. So this study is to detect DNA methylation proofs that are associated with insulin resistance and prospectively correlated with breast cancer development, both overall and in breast cancer subtypes among postmenopausal women, by conducting a population-level epigenome-wide association study. DNA methylation is tissue-specific, but the correlation between peripheral blood and tissue are gene-specific. So we first conducted a peripheral blood leukocyte-based EWAS study. and compared the methylation levels of detected CPGs with those of CPGs within breast cancer tissue and adjacent normal breast tissue. On the basis of the results in a published study of gene methylation parallelism between the peripheral blood cells and tissues in glucose metabolism, the peripheral blood leukocytes may be the best non-invasive alternative tissue standing for a surrogate DNA methylation marker that reflects multiple glucometabolic pathways. With the detected EOS-based insulin-resistant CPGs, we further tested for the associations with breast cancer risk in the peripheral blood leukocytes and conducted validation tests in breast cancer tissues. These allowed us to determine whether our insulin resistance CPG sites at genome-wide significance that are associated with breast cancer risk are systemic or tissue-specific or common in both. So the figure shows the diagram of study populations for EOS and the breast cancer study from the two cohorts. So WHI, the Women's Health Initiative cohort, this cohort is the largest prospective cohort study including the postmenopausal women. Their ages were 50 to 79 years at the time of enrollment between the 93 and 98 at more than 40 clinical centers in the US. For DNA methylation data, we included 3 WHI ancillary studies with available genome-wide DNA methylation measured in peripheral blood leukocytes, as shown in Figure 1, for the discovery AS315. For validation, BA23 and AS311 studies were included. For the purpose of our EOS analysis, we restricted our study population to those women who reported their race or ethnicity as non-Hispanic whites, a majority of the WHI ancillary studies population, and who had available insulin resistance phenotypes, including the fasting blood levels of glucose and insulin. These women had been followed up through March 6, 2021 with a mean of 17 years follow-up, and 80 of them have had developed invasive breast cancer. Our replication cohort was derived from TCGA breast cancer study. This study houses tissue-derived genome-wide DNA methylation data and molecular profiles of different breast cancer subtypes from breast cancer tissues. So we performed epigenome-wide DNA methylation array. In the WHI, we extracted DNA from peripheral blood leukocytes isolated from the fasting blood of the WHI participants, and the DNA methylation was measured by the Illumina 450 bits chip. We normalized methylation by BMIQ method. Leukocyte heterogeneities were estimated to be adjusted for in the analysis. TCGA, we obtained tissue-derived genome-wide DNA methylation data. DNA methylation was analyzed by Illumina 450K array, and we use MinFi-L software to normalize DNA methylation by normal exponential out-of-band background correction. We estimated the tumor purity and cell type proportions of each tumor sample. Okay, let's move on to results. we performed the epigenome-wide association analysis for the insulin resistance phenotypes. Among the 480,000 CPGs in the discovery data, we found several differentially methylated CPG sites associated with IL insulin resistance phenotypes, and further we validate them. So as you can see below two figures, these two figures showed the EWAS analysis results in the discovery as well as in the validation, particularly in association with the HOMA IL level. So in both line and scatter plots, we detected 35 CPGs at the genome-wide significance in the discovery, and 7 out of 35 CPGs were further validated. Next, we conducted a meta-analysis of all the detected epigenome-wide CPGs in relation to insulin-resistant phenotypes by combining their discovery and replication data. We detected several EWAS CPGs in multiple genes, including TXNIP, CPT1A, PHGDH, and ABCG1. Particularly, we detected one individual CPG, which is CG19693031, located in TXNIP gene, which was replicated inversely related to all insulin-resistant phenotypes at the epigenome-wide level. In the table, as you can see, this individual CPG had a negative association with the HOMA IL level at the genome-wide significance. Next, we conducted the association study of the detected insulin-resistant DNA methylation probes with breast cancer risk. So those replicated insulin-resistant genes, two genes, including CPT1A and ABCG1, were further correlated with breast cancer risk. As you can see below figures, in the CPT1A gene, breast cancer patients were associated with hypomethylation compared with non-breast cancer patients. In the ABCG1 gene, breast cancer tissues were associated with hypermethylation compared with normal adjacent breast cancer tissues. Particularly, only one individual CPG, which is CG016966, I'm sorry, it's a 01676795, located in the POL gene, was commonly detected across the two cohorts. So these CPG with one standardized deviation increase in DNA methylation has a 75% increase risk for developing breast cancer, particularly for the hormone receptor positive subtypes in WHI. This CPG also has had 5 times greater risk of developing breast cancer in the TCGA cohort. Further, we compared the DNA methylation levels between the WHI and TCGA using the insulin resistant CPGs associated with breast cancer that are shared by the two cohorts in terms of gene region, CPG context, and enhancer. The methylation levels of some CPG context and gene regions differed significantly between the cohorts among the non-breast cancer subcohorts. However, no significant difference in methylation levels between the cohorts was observed within the breast cancer subcohorts. These suggested DNA methylation parallelism between peripheral blood leukocytes and tissues in terms of breast cancer. So conclusion, this is the first large population level EWA study conducted among postmenopausal women for detecting differentially methylated CpG sites in the peripheral blood leukocytes that are associated with insulin-resistant phenotypes and further prospectively associated with breast cancer development both overall and in breast cancer molecular subtypes. The methylation levels of the detected CpGs in insulin resistance and breast cancer risk between the peripheral blood leukocytes and the breast cancer tissues were comparable. So this finding is consistent with the previous findings of the gene methylation parallelism study in glucose metabolism. So these suggest that the peripheral blood leukocytes may serve as the best source of surrogate DNA methylation markers in non-invasive tissues, reflecting multiple interconnected glucometabolic carcinogenesis pathways. So in conclusion, we found several differentially methylated CpGs which are both well established and novel at the genome-wide significance in relation to insulin resistance that are further correlated with breast cancer development. So our study findings contribute to better understanding of the interconnected molecular pathways on the DNA methylome between glucose intolerance and breast cancer carcinogenesis. So our findings suggest the potential use of DNA methylation markers in peripheral blood leukocytes as preventive targets for detecting the group for breast cancer at risk among postmenopausal women.